My name is Katie Galloway. I'm an assistant professor in the Department of Chemical Engineering at MIT. I am not Herbert Levine. Herbert was prevented from being here and being today's session chair by, I believe, some illness, and so we're hoping he recovers well. But it's my pleasure to get to introduce the speakers for the session on condensates and genome architecture. And we're going to get to start with Ben Zhang, an associate professor of chemistry at MIT. His lab aims to build a global framework of the human genome, connecting its sequence and structure to activity. And we're looking forward to hearing him kick us off here. Right. Thank you.
Oh, can you hear me? OK. All right. Well, thank you, Katie. And well, thanks the organizer again for the opportunity to speak. So yes, as Katie mentioned, my lab focuses on computational modeling of genome organization. And I guess for this audience, I don't really have to dwell on the importance of the three-dimensional genome structure. But I just want to point out that as you all appreciate, the genome organization is actually quite complicated, and it's intrinsically a multi-scale problem. And because of this complexity, we try to study the genome structure at a different scale with different approaches. On the one hand, we try to study the whole genome organization using integrative approach that we combine modeling with experimental data. On the other hand, we also try to study the structure at more or less atomistic level, but for a much smaller region with what we call the near atomistic modeling. So we are really kind of using this multi-scale approach. But I guess in multi-scale modeling nowadays, it's kind of like a buzzword that gets thrown around a lot, right? But then still back to the question, how are we going to sort of study this problem, such a complex system with computers, right? I don't really do high throughput sequencing, and we don't really have a microscope. So the technique that we rely on is called molecular dynamic simulations. It was pioneered by this 2013 Nobel laureate for biomolecules. So the idea that you can essentially approximate the interaction among all the atoms that are dictated by quantum mechanics with the empirical energy function or force field. And once we have this energy function, we can then essentially solve Newton's equation to predict structure and to study the dynamics. And this sort of energy landscape focused perspective has proven to be very powerful for our understanding of biomolecular and most notably protein. And some of the pioneers who proposed this idea for protein folding are actually here today, including Peter Wallinis and Dave Turumolai. So I guess, but there is some technical difficulty though when we try to directly apply the Nobel-winning idea to chromatin. And the problem is that the so-called near all-atom force field, they are very accurate. They have all the details for every single atom or the water molecule count ions, but they are also highly expensive because of this detail. Even for just a single nucleosome, we are kind of limited by to the time scale of about a microsecond, and so then it's really hard to generalize that to a long chromatin, then of course not the whole genome. So then to study really much larger scale question, we actually have been trying to we have to develop basically approximate but accurate energy function. That's actually kind of the unifying theme of my lab. We try to sort of invent new coarse-grained model, but accurate models to really that allow us to study the structure and the dynamics of the genome. And today I'm going to give you two examples so explain sort of how do we do that at different scales. Okay, and we're going to start with the near atomistic model. So there, before I get into detail about the model, I do want to sort of motivate this a little bit. Our question there is actually essentially quite simple. We just want to understand, what exactly is the structure for just a string of nucleosome? Well, it turns out, the answer to that question is actually quite controversial. So for many labs actually have tried to study this problem, and the so-called in vitro study where when the chromatin was taken outside of the nucleus, and it was often found that chromatin can sort of form this kind of very regular fiber-like structures, and also have sort of led to the atomic model where you can see this sort of clear sort of kind of two helix-like fiber structure. But unfortunately, in the meantime, many labs have also tried to study the structure in vivo inside of the nucleus, and there, unfortunately, this fiber structure was never really able to be found. And this was kind of the latest example from the Kornberg lab, which provided really remarkable resolution where they studied the mitotic chromosome structure with the cryo-electron tomography, and you see that there's just kind of seem to be a disordered mass. There doesn't seem to be any sort of structural altering. And another recent sort of exciting development in the area is the apparent liquid-liquid phase separation over chromatin segments, and but it's sort of still unclear whether those condensates are really more like a liquid or they are gel, right? So our goal here is that we want to use the computational modeling maybe to provide more insight into the stability of the different conformations and to see whether there is any connection between in vitro or any in vivo structure, and how do different conformations actually contribute to the different sort of phase or dynamics of the condensates. Okay, so as I mentioned that the atomistic model really does not provide sort of the time scale that's needed to study chromatin structure, chromatin organization. So we actually rely on, we call this near atomistic model, where we use sort of implicit solvent model and removing water molecules sort of significantly reduce the system size. And we also use fewer sort of particles for the DNA and for the protein. And these are actually rather standard approximation actually has been adopted by many labs. But here what's sort of crucial is that we does sort of provide the chemical identity of individual amino acids and individual nucleotides, and that allows us to to use essentially a more physical chemistry potential to describe the interaction among the protein, among the DNA, and a lot of the interaction are actually sort of very well defined, has been derived based on rigorous statistical mechanics series, and we can also account for the effect of the solvation of water molecules sort of implicitly. Now this model, of course, not perfect. No model is perfect, and we're well aware of the limitations. So we actually have also been trying to sort of develop new methods to improve the accuracy of those models, and in particular, we focus on sort of trying to develop a protein model that can sort of consistently describe both order and disorder protein, and they have heard about in many talks. So those proteins are actually very important for phase separation. So it's but in a sort of really achieving a consistent accuracy for both order and disorder domain of those proteins turned out to be actually quite challenging. So we actually made a lot of progress in ensuring the accuracy of those coarse-grained models. And recently we have been sort of applying deep learning idea to further sort of improve the optimization of those models and sort of improving the simulation efficiencies. All right, so now with all those sort of technical issues sort of addressed, we're ready to study chromatin folding. Now, one nucleosome at a time, though. So here, we looked at basically the unfolding of a single nucleosome. Essentially, we are looking at the unwrapping of the DNA molecule from the histone protein. So here, the DNA is showing gold, and the histone protein are showing sort of in the rainbow colors. So we're basically looking at the end-to-end distance of the DNA, and we're computing the free energy cost for sort of essentially unwrapping the DNA from the histone protein. So this is the crystal structure. Remember, in the sort of crystal structure, the DNA is fully wrapped for about 2 rounds around over the histone proteins. So what you see here is that the free energy essentially sort of increases more or less monotonically as the DNA unwraps from the histone protein, which is reasonable because we know that the DNA is actually negatively charged, and the histone protein is actually positively charged. So the complex is actually quite stable, and it's stabilized by electrostatic interactions. But now if we look into more detail, we actually find something quite interesting. So what you see that this free energy curve can more or less sort of separate into 3 regimes. There are essentially 2 low force regimes. So here force corresponds to the gradient of the energy over the distance. So there are these 2 low force regimes separated by a high force regime. And interestingly, those 2 low force regimes actually correspond to exactly the unwrapping of the outer and inner layer of the nucleosome. Just a reminder, the DNA wraps for about 2 rounds. And then, the presence of this high barrier essentially decouple the unwrapping of outer and inner layer DNA. And that's actually quite important. It sort of explains how nucleosome can really balance stability versus flexibility. So the idea that if you look at it, remember, once the nucleosome sort of is fully wrapped, then essentially there is no way for other protein to access the DNA. So then in order to function, the nucleosome had to somehow open up. And what you can see that here, unwrapping the outer layer actually only incur a very minimal sort of energetic cost. So it could happen over a second time scale. It has been shown by many experiments. On the other hand, unwrapping the inner layer is actually energetically prohibitive because of the presence of this barrier. And that's important because once you sort of start to unwrap the inner layer, then you start to lead to irreversible nucleosome loss. So that actually can lead to genome instability. And this 3 regime is actually consistent with the single molecule experiment performed from the Michelle Wang lab, and the numbers we get are also actually in quantitative agreement with the single molecule measurements. And importantly, because we do molecular simulation, we actually have the resolution to really understand, what's given rise to that barrier that sort of decouple the unwrapping of outer and inner nucleosome layer. And in the end, we found that it's actually really the distal region of the histone protein. They can sort of dynamically move around to try to hold on to the DNA for as long as possible. I'm not going to go into too much detail into how we arrive at that conclusion, but I do want to highlight that, basically, use this as an example to highlight the usefulness of this kind of near atomistic model. So they are actually accurate enough to allow quantitative comparison with the single molecule experiments. And in the meantime, they also have the structured detail to really provide molecular insight into and to explain a lot of the experimental observations. Okay? All right, so now, encouraged by our single nucleosome study, we're ready to, we get more ambitious, so we next want to look at a tetranucleosome. So this is a crystal structure for the tetranucleosome. So here you sort of start to see this this fiber configuration, right? You sort of have this 2 columns of nucleosomes. And this system now is complex enough that actually allow us to sort of ask an interesting question, right? We can ask sort of, and we sort of kind of broadly cluster them as the chromatin folding questions. For example, what are the possible conformations that can be adopted by this tetranucleosome beyond maybe just this single crystal structure? And what are maybe the key degree of freedom that sort of dictate the conformation change of the tetranucleosome, and how does the different chemical modification and changing in link length, how do they affect the stability of the structures? Here, for this tetranucleosome in our sort of corresponding model, it only has about 8,000 particles, which is actually rather small by the standard of nowadays computing powers. But even for this system, we really cannot hope that we'll just sort of run brute force simulation for a long time and we'll solve all the chromatin folding problem. And the reason is that, as I mentioned, in a single nucleosome study, so the nucleosome is actually stabilized by the electrostatic interaction between protein and DNA. And now, in this system, those are sort of non-specific but very strong interactions, they also contribute to the inter-nucleosome contact, inter-nucleosome interactions. And so those very strong but non-specific interactions, they sort of can actually give you sort of slow kinetics and a lot of sort of kinetic traps. But what all of that means is that, basically, if we want to sort of look at larger scale conformation changes, we have to be sort of smart. We look at a lot of sort of simulation techniques, and in the end, we were actually able to sort of found a technique that actually works quite well. We were actually able to sort of study chromatin folding and unfolding in a single trajectory continuously. So here, where I'm showing you the radiated gyration that kind of measures the overall shape of the chromatin, which you see that the chromatin expand, collapse multiple times. Okay. And if we look at the specific context among the nucleosome, we saw that the contact form, that get disrupted, they form again. Okay, so we can really sort of start to see this sort of folding and unfolding transition in a single trajectory. Now, then what we did was basically run many of those simulations, and we started to watch movies. That's kind of part of the fun, actually, being a computational modeler. We can sort of generate all these very interesting, cute movies we can sort of look at and try to understand things. So interestingly, we actually found 2 very distinct pathways for chromatin folding. So in this we call a concerted pathway, we found that the contacts more or less form on similar time scales. So here, in our simulation, we do have all the histone protein. I'm not showing them just for clarity. Then in this sequential pathway, we found that a subset of the contact form first, and then you sort of see this kind of trinucleosome motif followed by the other ones. Okay, now the difference between those two movies are probably better explained with this sort of two-dimensional projection. So here, remember that the tetranucleosome has a more or less a zigzag configuration. So you have in the PDB structure, you have 1, 3 on top of each other, and 2, 4 on top of each other. So here, I'm just quantifying the distance between the 1, 3, and 2, 4 nucleosomes. And the PDB basically more or less localized at this region where those two distances are very small. So the blue dots are essentially the scatter of all of our simulation, which is a quite comprehensive sort of sampling. And the yellow and the pink line correspond to the two movies I showed you in the previous slide. You can see that along the yellow trajectory, the two distances are actually quite correlated with each other. That's why we call this concerted. And then along the pink trajectory, we actually see that one of the distance collapse first, followed by the other one, right? Why this is interesting? Well, I guess, before I explain why this is interesting, it's actually quite exciting because recently we found this paper from the Kramer lab where they use cryo-EM to actually sort of directly solve this trinucleosome structure, which actually looks exactly like what we predicted by along this sequential pathway. So, so that this trinucleosome structure actually might be actually quite relevant as a metastable state. Now, what's really exciting, I think, is that those intermediate structures we see along those folding pathways, they are actually also seen in vivo, in the nucleus. In particular, so those kind of trinucleosome structure actually has been reported, in many studies where when they try to directly visualize the chromatin in the nucleus. And in the meantime, we found that along our sort of sequential trajectory, we also sort of see this, they call this beta rumba structure that has been reported in the Cell paper where they try to use high-C to study chromatin structure. So then this kind of raises a very interesting hypothesis, which is that maybe a lot of those in vivo structures that are not really that random. They may appear irregular because they are actually more like folding intermediates or unfolded excited state, if you like. Maybe the fiber structure is actually indeed quite connected with them, but maybe just a change of the condition that somehow drive the chromatin unfolding from the fiber structure, which will result in those sort of in vivo configurations. Okay, well, I guess but one question that one sort of technical issue is that when we were running those simulations, we were using sort of biased samplings, and so we figure out a very sort of complicated way to address that, which basically allow us to quantify the stability of every sort of single chromatin structure without any biases. And what's really useful is that now we can actually use those free energy profile to compute the most probable transition path without any bias. And again, we sort of show that the folding indeed occurs sort of through this both concerted and sequential pathway, and the intermediate state it does resemble the one we see in the in vivo configurations. And interestingly, we found that when we sort of start to introduce histone modifications, which are frequently found in vivo, they actually also tend to stabilize the in vivo configuration as well. And that, again, sort of supports the idea that maybe that's why we see those sort of in vivo configuration more often, those irregular configuration more often, because now the energy landscape is kind of more tilted to favor those structures. Okay. All right. Yeah. So what I just told you is a tetranucleosome, right? It's kind of cute, but it's not really a chromatin. So then we wondered, how does the findings sort of how general are they, right? So then to test that idea, we now look at 12-mer, which you can sort of now clearly see this kind of fiber configurations. And basically then to facilitate unfolding, we apply a tension at the two ends. Then we look at a different mode for chromatin unfolding, and we can compute a free energy profile. And from those calculations, we can basically compute a force extension curve. And from those force extension curves, we can then we can also compare with the single molecule experiment. I would say that the agreement actually is quite reasonable, especially considering we use a slightly different salt concentration. But what's really exciting is that our simulation also captures those two clear regimes. There's this linear regime where a larger increase in force really sort of only incur a very minor increase of the extension of the chromatin. Then you have this plateau region where a small perturbation in the force leads to a huge change in the chromatin. Okay? But what's really, really exciting to me was that when we start to look at the intermediate structures at sort of pulling forces, we actually never see this sort of a uniform extension of chromatin. And this was kind of kind of was proposed before, because if you look at this structure, it's very symmetric, right? It's really kind of like a Hookean spring. So if you pull on a Hookean spring, you would expect more or less expect a uniform extension. But that was actually not really happening at all in our simulation. Instead, we found that the chromatin tend to break into small clusters, and those small cluster structure are actually quite similar to what we found in the Tetranucleosome simulation. Now, these structures are actually very, very exciting because recently many labs use super resolution imaging to look at the nucleosome structure in the nucleus, and they reported that the chromatin in vivo indeed form those kind of nucleosome clutches. So that's actually sort of quite similar to what we found for the structure in our simulation. So again, this kind of supports the idea that maybe there is indeed a connection between sort of this fiber structure with the in vivo configuration. And the force we see the chromatin unfolding is actually quite reasonable. It's only about 3 to 4 piconewton, which are actually quite expected for molecular motor or for the RNA polymerase. Okay, so right, so extension, I would say tension or force is one sort of variable that's frequently encountered by chromatin. Another important factor for chromatin is that in the nucleus is that it's never in isolation, right? It's always in a crowded environment, maybe surrounded by other chromatins. So then we wonder, how does the crowding affect the chromatin structure? To answer that question, we now look at again chromatin unfolding, but with the presence of another chromatin segment. So we're now looking at the simulation with 2 chromatin segments. And this acts to quantify the contacts between the 2 chromatin. You see that as they form more contacts, the free energy decreases, so meaning that they're actually favorable for their binding. So this y-axis sort of quantifies, the extension of the chromatin, the unfolding of the chromatin. So then what's really interesting, again, is that when we start to look at the conformation for the bound chromatin segments, this is probably not too surprising. We found that in one of the configurations that the 2 chromatin more or less sort of stay in their fiber structure, and they sort of come in close contact with a very weak interaction mediated by histone tails, and this binding favorability sort of is consistent with the idea with the experiment from the Rosen lab where they show that the chromatin can sort of spontaneously form liquid droplets, right? But then what's really exciting is that we've also found an alternative binding mode that has quite comparable free energy, but now they have very different structures. So here now, what you see that the chromatin actually they start to sort of unfold, so the extension of each individual chromatin by itself is not favorable. But now this unfolding actually essentially frees the nucleosome to allow them to sort of adopt many different orientations that actually promote the interdigitation across chains. So they actually allow the chain to form more contact and those sort of and also form stronger interaction, and that actually sort of then essentially compensates for the penalty of extending individual chromatin. And this now the formation of a stronger interaction and also the possibility of forming topological entanglement here because they're more extended. Actually, it will be consistent with the observation that the chromatin, some of the contents are actually really more like a gel instead of liquid. Remarkably, I was actually at this other meeting last week, and then I realized that there's this paper where they sort of again, this from the Kornberg lab where they sort of look at the chromatin structure directly in nucleus with remarkable resolution, and you see that they indeed see a lot of those interdigitation that are quite similar to what we see in our simulations. All right, so yeah, so just to conclude this part, I guess I was trained in the Walden's lab, and we'd like to think with the energy landscape. So just to summarize this part, we believe that if I draw a cartoon energy landscape, we believe that the fiber structure is still maybe the global minimum, but the fiber structure maybe is not that much more super stable compared to some of the irregular chromatin configurations. So then a lot of those in vivo factors like tension, crowding, histone modification, they can essentially tilt the balance, and which then basically drive the unfolding of the fiber and give rise to those sort of irregular structure. And once the chromatin become unfold, they can sort of easily sort of promote multivalent interaction, sort of start to form those condensates. Okay. All right, and and in addition to studying bare chromatin, we have also been looking into some of the chromatin regulators and how do they sort of form condensate and how do those condensates sort of affect the chromatin stability. But I'm going to skip all of those, because I want to really sort of start to talk about the whole genome architecture, right? Because all the structure that I have showed you so far, they're really at the bottom of this diagram, which only about 10,000 base pairs. I mean, if we try hard enough, maybe we can work sort of to 100 kilo base pair. But that's really sort of far away from the 6 billion base pair of the entire human genome, right? So then to study the structure of the entire human genome, we really have to sort of essentially throw away our beloved atomistic detail and work with coarse-grained models. And so this is essentially a cartoon representation of our. We do have a 3D model. This is a 2D cartoon. So here we include every sort of one of the 46 chromosomes of the human nucleus, and we represent each chromosome as a sort of particle with a bead on a string model. So this particle-based representation, just to be clear, now it actually allows us again to basically to perform molecular dynamic simulation. So the technique that we use at this scale is essentially the same as we had before. But one sort of a key difference is that because we are working with the coarse-grained model, we really don't have the luxury to sort of lean on all those decades of research, and we don't have an energy function. And that's kind of the first thing we have to do. We have to sort of basically, come up with an energy function that actually sort of describe the entire human genome. So it just gives. So this is a very simple illustration of our energy function, which is sort of account for the connectivity of the polymers, and we also have an energy term that approximates the effect of a compartment and the effect of a loop extrusion. And now, what the design of the energy function itself is actually a very interesting story, but I won't go into too much details. Now, but once we settle on the energy function, we still have to parameterize the energy function. We still have to figure out the interaction strength of the different particles in the system, right? That's important for sort of predicting the stability of the structure for ensuring the biological relevance of our structures. Again, here I would say coarse-graining is actually a very popular idea in the theoretical chemistry community, but we really cannot just rely on existing approaches because, as I mentioned, each one of our bead actually corresponds to 1 million base pairs. So this the complexity of the chemical composition of the bead and also the larger size essentially render the very popular bottom-up sort of parameterization approach inevitable. We simply cannot afford to run just maybe quantum mechanical calculation and try to figure out the interaction among the particles in our system. So instead, to sort of parameterize the energy function, we really have to think outside the box. So that's where the high C comes in. I guess after y'all's talk, you all know sort of high C. I'm not going to go into any of the details. So this technique would actually sort of introduce when I was working at the postdoc with Peter Wallendes. So this diagram basically sort of explains more or less how this technique works. So we start with an energy function. Then with this energy function, we can basically then perform molecular dynamic simulation to produce an ensemble of structures. And from this ensemble of structures, we can then compute an in silico contact map. Then we can basically compare the difference between the in silico map with the high C map. We can then use their difference to basically sort of update the parameter in the energy function. And we can do this iteratively so that in the end, we are able to arrive at an energy function and an ensemble of structure that reproduce experimental data. Okay. All right. So I'm going to skip that. So then but I guess after we obtain the structure, we sort of carry out extensive validations, and it's probably not too surprising. We were actually able to reproduce high-C data, given that it was kind of provided for model training. So this is the experiment, this is the simulation, and here basically the zoom in of different regions of the contact map. We also have sort of sort of quantitative metric to convince ourselves we're actually having quite good agreement with the experiment. But importantly, our model was also able to reproduce a variety of sort of imaging results as well. So I guess so chromosomes are actually known to form territory. There's not a lot of intermingling among the chromosomes, and you can clearly see that from our representative structure as well. So here we identify each chromosome with an individual color. You can see that this is basically an expansion of that structure. And you can see that the chromosomes are more or less collapsed. There's not really much of a mingling among them. And now if we now color the chromatin by the types, you can clearly see that there is a phase separation between heterochromatin and the euchromatin, where the heterochromatin is shown in blue, and also the heterochromatin typically more or less localized at the periphery of the structure. And when we look at now the radial position of individual chromosomes in the nucleus, again, we sort of see a very nice correlation with the experimental measurements. So again, I want to emphasize that none of those imaging results were actually provided for model training, right? So the model was purely based on high-C data. So the fact that we were able to, recover all those results, again, I would say provide confidence, in both the modeling strategy and also in our simulated structures. All right. Well, so you might ask, right? So so what? You can get a structure. What can we learn from the structure that we're not already known from the high-C data, right? Well, one thing I would say for the advantage of having structure is that they're actually very useful for building intuitions. So here you can clearly see that from this collaboration with the Brad Bernstein lab at the Broad. The Bernstein lab, they generate high-C data for both tumor and normal samples. And so here you see that, right, so this is the tumor high-C data. This is normal high-C data, this is tumor high-C data. If you just look at the high-C, you can see some changes. Here this is a little bit wider, meaning they have a lower frequency, contact frequency, but it's hard to conceptualize what exactly does that mean in terms of the 3D structure, right? But I hope you can appreciate the larger scale change we see, from our simulations. So here we're again coloring the genome structure by the types. So in normal sample, you see the canonical picture where the heterochromatin more or less localized at the periphery, and the euchromatin stayed in the interior. But in tumor, you see that there's this large-scale reorganization where now the heterochromatin actually start to move more towards the interior of the nucleus. So when I first saw this, we were actually really excited because we sort of proposed that this change might actually sort of be underlying some of the change in gene expression of tumors. So we're very excited. We submitted the manuscript, but of course, it's only fair that we encountered reviewer number 3 who were not very impressed, right? And they were like, these are just sort of in silico predictions. Why should they care? Honestly, when I sort of, I kind of secretly agree with the reviewer 3 a little bit because it was a model, right? And a lot could go wrong in the model, and so but the good thing is that we have excellent collaborators, and they actually then went back to the lab and performed EM images on the 2 cell types, and lo and behold, they found exactly the same thing as we see in our simulations. So here in its images, the heterochromatin are showing darker region. So you see that in the normal cells, the heterochromatin does look like at the periphery, but now in tumor, you see that indeed a lot of the heterochromatin start to dissociate and toward the interior. And and then other sort of prediction on the centromere cluster were also consistent with the previous imaging as well. All right, so over the years, we have also looked at the topology of the chromosome, and we found that, I guess it's kind of in line with Yob's talk, we found that the interface of chromosomes are indeed actually free of topological entanglements, and we found that for mitotic chromosomes, there actually is a very sort of interesting kind of very long and hierarchy, so this kind of helical ordering, which also seems to be in line with the Yob's high-C data as well. All right, so So yeah, so I mean, in addition to sort of generating structures, I would say that our energy landscape-based approach is actually very useful for studying dynamics, which I think is kind of a key sort of advantage of this type of approach. And that sort of dynamical framework is ideally suited to actually study a lot of the questions regarding the condensate in the nucleus. Just as a reminder, inside of the nucleus, in addition to DNA, there are also the nuclear bodies, including nucleoli and the speckles, and they're very important for RNA processing, for gene transcription. And as we sort of already heard many talks, that those nuclear bodies, they're kind of like a liquid droplets that can fuse when they merge. And this idea also seems to be quite general, as been reviewed from, in the paper from the Young lab, where this condensate might actually underlie the transcription and heterochromatin formation. So it's a very intuitive idea, and sort of it's consistent with a lot of experiment and generated a lot of excitement, understandably, right? But not to ruin the party, I would say phase separation does not really explain everything, which is I guess probably true for every model or every theory. So one thing that puzzled us for a little bit was that so there is the apparent coexistence of multiple of those nuclear bodies, right? So these are basically the images of the nucleus where now the cells are stained with antibody to highlight the nucleoli. You can clearly see multiple of nucleoli in each individual nucleus, and this is basically a quantification of the image shown on the left, right? So I guess we all heard from the Ibrahim's talk where the phase separation should sort of more or less follow a classical nucleation theory. But the classical nucleation theory also predicts that the thermodynamic equilibrium really should just consist of one single droplet to minimize the surface tension. So then the other question is that why what's so unique about the environment in the nucleus that's preventing the coarsening of those droplets, right? Preventing the coarsening of all the nuclear body into one single droplet. Well, there could be many explanations. As Arup sort of mentioned, it could be that some of the non-equilibrium reaction and the RNA production. But we wondered that could the topology or the organization of the chromosome also contribute to this to the coarsening of those droplets, right? So now we have a reliable model for the genome. We're actually quite well positioned to address that question. So basically, to study phase separation in the nucleus, what we did, we just introduced additional protein particles. I know sort of thinking of those spherical particles as sort of several hundred bit amino acids sort of takes a little bit imagination, but we're also just after sort of qualitative pictures, so I think this actually works quite well. But we were careful with about the sort of the size, the concentration of those particles. We basically choose those parameters to model the nucleolar protein that are sort of driving the formation of a nucleoli. Okay, and we sort of we also introduce a specific interaction between those proteins and the specific region of a chromatin that are known to bond with the nucleoli. And then we also introduce a specific interaction among those proteins to basically drive their phase separation. And I'm just going to show you a movie to sort of give you some idea how does everything works. So this is essentially our initial configuration where we start with a random distribution of the protein particles. And as you can see that as time evolves, we see that the proteins start to sort of aggregate because they have favorable interactions. And you sort of see that the formation of a small cluster, and small cluster eventually sort of merge with a larger cluster. We can also track the number of clusters as a function of time. You can see the initial burst and sort of followed by a coarsening. Very interestingly, in the end of the simulation, as you see here, we actually do not end up with one single droplet. Instead, we actually end up with two droplets. Okay. And now this is, again, inconsistent with maybe a naive prediction of the classical nucleation theory, but it's consistent with biology, right? As we sort of see from the previous images, cells typically have multiple of those droplets. Now, of course, I only showed you just one movie, and we sort of performed many more simulations, and we found that in all but one simulation, we found most of our sort of system actually end up with this multiple droplet state. Now, so that's actually getting more interesting. So, at least our observation is statistically significant. Now, before we get too excited, we also sort of try to compare more with some of the kinetic measurements of phase separation in the nucleus. This is an experiment from the Brangwyn lab where they directly monitor the coarsening kinetics of droplet formation in the nucleus. They basically quantify the average size of the droplet as a function of time. They found a Pavlov behavior with an exponent of 0.1. So the exponent itself is actually quite interesting because it deviated from the sort of the more classical value of 0.3. But we found our simulation actually was able to reproduce that quite well. And this is another experiment from the Lewicka lab where they directly look at the coarsening kinetics of two fusion nucleoli, and they quantify the average the neck radius as a function of time, and sort of say this Pavlov behavior with the exponent of 0.2, which was actually also reproduced in our simulations. Now, as another check, we sort of went back to our tumor samples. So here we basically carry out a phase separation simulation in tumors. So everything regarding all this protein-protein interaction, protein-chromatin interaction, kept the same. So the only thing we changed here was the interaction among the chromosomes, which are now derived from the tumor high-C data. So they previously were derived from the normal high-C data. And interestingly, that change is actually sufficient to drive the formation of more of those droplets. So here we see a clear increase in the number of droplets in tumor samples. And that's actually very interesting because for tumors, we actually know that they tend to have more of those droplets. So that the fact that our model was actually able to reproduce that trend, at least qualitatively, is quite reassuring. So, of course, in the end, our goal was not to reproduce something that you guys already know. The goal is to see whether we can learn something new from the model, right? So then we wonder, why are we why our simulation are predicting more droplets than just a single droplets? So then, as a chemist, I like to think this as a chemical reaction. So we define a reaction coordinate, which essentially quantifies the distance between the two droplets, right? So at a larger distance, you sort of see there's two droplets, and at this small distance, you have one droplet. And we then basically compute a free energy profile. And interestingly, we actually see a barrier that at the point where the two droplets start to touch each other. And this barrier, it's not expected from, again, a naive sort of classical nucleation theory, but what's really surprising that the barrier is also mostly driven by entropy. We can sort of do some tricks of free energy decomposition. So we sort of scratch our head a little bit, and in the end, we figure that there is probably actually a very simple explanation for this barrier. And what's happening is that so because we are sort of looking at the simulation, looking at the phase separation in a network of chromatins, and because there are sort of a favorable interaction between the droplet and the chromatin, so those droplets are essentially tethered to the chromatin network. So then, as the droplets they try to move close to each other, they try to merge, they essentially have to pull on the chromatin network. And then the chromatin, I mean, in a very sort of simplified version, you can sort of think of them as more like a rubber. Like any polymer network, they're kind of like a rubber. If you pull on the rubber, there will be a countering force, right? And that countering force is what's giving rise to this barrier. And also, because of the rubber are really kind of like entropic springs, they explain the entropic origin in our simulations. Okay. All right. So yeah. So far, what I told you is that the chromatin can actually have a big impact on the kinetics of phase separation, on the kinetics of those droplet formation. So then, but we wonder, can the droplet actually also affect the dynamics of the chromatin, right? So that actually turned out to be quite apparent in our latest venture, where we try to now build a more complete model for the nucleus. So here, in addition to chromosome and the nucleoli, we also introduced speckle, which is another key nuclear body, and we also included the nuclear lamina. And so in this model, not only were we able to reproduce high C, but also the contact between the chromatin and all these different nuclear landmarks. So this should provide a more accurate model for the genome and the nucleus. but I guess you might wonder that as we sort of start to introduce more and more complexity, I mean, can we even can we actually learn anything at all from this very complex system? Well, it turned out actually a very simple picture started to emerge from our simulation. And in a very simple model, I can actually explain a lot of those experimental data about the chromosome contact and the interaction between chromosome and the nuclear landmarks. And so the model we currently have is that there is essentially a simple sort of a co-phase separation between the chromosome and the nuclear landmark. So the idea that if you start from this more or less disordered structures, so then the specific molecules they could actually nucleate at a specific region of the chromatin, and those nucleations essentially drive the formation of those nuclear bodies. And this co-phase separation model, I would say, actually it can explain two almost contradictory picture of the genome organization. Because on the one hand, so now the nucleation basically explain that a lot of the nuclear body, they are essentially arrested because they're tethered to the chromatin. They really cannot move over a larger distance, and that's actually quite consistent with some of the experiments from the Belmont lab where they look at the time trace of the speckles. They sort of see that their movement are actually really constrained. And the nucleation also explains that how would those nuclear bodies form very precise and specific contact with a set of chromatin segments. And so this is actually analysis of the single cell data from the Zhang lab. So here we're looking at the chromosome region that are contacting speckles. And so this is the genomic region, and this is the single cells. You can see that there are clear, so the orange dot means they are in contact. You can clearly see that there are a subset of chromatin regions that are essentially always in contact with the speckle. And the distance actually are very precise. It's always about sort of within 300 nanometers. So then this, and sort of this nucleation would actually explain that, how that would have sort of formed those potentially functionally important contacts. But this co-phase separation also explains another sort of a behavior which has been known for a long time, which is that the overall organization of the genome is actually quite heterogeneous and quite sort of random almost. And the idea that because the phase separation is non-specific, right? In different cells, those nuclear bodies could be engaging with a different set of chromatin that are forming those contacts, and this heterogeneity in the contact can essentially drive the heterogeneity in the chromosome position and in the overall organization of the genome. Okay, so we believe that this sort of simple model can actually reconcile how you would end up with a sort of globally random picture, but locally you have very precise contacts that are probably important for function. All right, so and in addition to study the whole nucleus with the experimental data, we have also been trying to predict the Hi-C data from chromatin states, but I won't get into all of the detail, but instead I want to acknowledge people who actually did the work. I was fortunate to work with an excellent group of people, and this picture was clearly taken before COVID, where when close contact is still thin. And we are trying to sort of update the picture. But it's, so a lot of the work was actually done by a very talented PhD student, Yifan, and also Xinchen and Shumin. And I want to thank the collaborators and my funding, and thank you for your attention.
Thanks for the interesting talk. I have a question about the droplet. Like you mentioned, through simulation, there are finally two droplets forming, like small droplets merge, right. And in contrast to the classical nucleation theory. Right. And the reason or the explanation you gave is that they have a higher energy barrier for it to finally merge into one, and right.
Free energy barrier, yeah.
Yeah, and there are strings attached to them. Then I wonder why, in the beginning, those small droplets, they will merge, finally to two droplets.
Right, that's a great question. So, so the coarsening of the droplet, they can sort of go through typically two mechanisms. One mechanism is they call it also ripening, where the droplet that will actually just sort of essentially vaporize so that the molecule can move to other droplets. So I think that's what actually a lot of what's happening initially at the beginning, how the small cluster merge. The small cluster will just disappear because they sort of have a small free energy penalty for them to disappear. So they will actually disappear and merge with the other droplets. But as the droplets sort of grow larger and larger, then the penalty for them to just completely vaporize is actually much larger. So that's why they will only merge when they sort of through this Brownian diffusion, when they sort of get close to each other, they will actually merge. And so the barrier we sort of see is that there will actually be a barrier for them to really sort of to get close to each other. That's why they actually can significantly slow down the coarsening.
Hi. Hi. A similar question. So, does that mean the number of condensates, the number of droplets after at the steady state, or like after a while, does that is it dependent on the size of nucleus, and is it like, I mean, I don't know anything about the actual biology of the droplets, but is it, I mean, if it's actually in the system, in your model, if it's actually dependent on the size of nucleus, is it representative of the actual biology?
Right. No, that's a great point. So, okay. I mean, I showed you the result where we see that in tumor, right? We sort of see more of those droplets, and in our model we didn't change the nucleus size, but what happened is that there are weaker interaction among the chromosome, and which sort of essentially make it even more unfavorable for them to really get close to each other. But I think you're right. I mean, in a more realistic sort of a tumor model, it's probably because they have a larger nucleus size, and that's why sort of they have weaker contacts, and that sort of further gives more penalty for them to really merge each other to form a single droplet. Yeah.
Thank you.
Yeah.
Hi, Ben.
Yeah.
Interesting talk. So I had a question about the whole genome simulations where you show different chromosomes are at a different radial position. Mm-hmm. So I was wondering, can you tell us a little bit about how you start the simulation? And if you randomize the position, would they sort of reach those kinds of radial positioning deterministically or with some reliability? Or is it more random?
Are you referring to this plot?
Yeah. Yeah, so
that's a great question. So here actually we are plotting essentially an average over many simulations. Yeah, so we always sort of start from just kind of random positions, and I mean on average you sort of see this clear trend where some of the active chromosomes tend to be at the interior, sort of inactive ones tend to be at the periphery. But if you look at it sort of individual simulation, individual cells, I would say there's actually quite a lot of fluctuation and heterogeneity. I mean, that's also sort of been known that, if you look at the single cell data from the Zhang lab, where you can clearly see that there are huge fluctuation over the position of a chromosome. But on average, you start the trend emerges. Yeah.
I see. So do you see them like the movement from, let's say, interior to the periphery for a chromosome, one of the bigger chromosomes, or X or 8? I mean, if you start it from the middle, in a single simulation, do you see it move, or you really it's a phenomenon that's like derived from the ensemble that they are more likely to be outside because they are bigger?
So in our simulation, I mean, we do sort of see, the simulation result really does not sort of depend on the initial condition. I guess we are sort of using effective equilibrium model. So in the end, we were able to sort of reach a thermodynamic equilibrium, and we sort of get this converged result. But I mean, in reality, I mean, it is possible that the system does not fully equilibrate. So maybe that the final position of the chromosome would depend a little bit on sort of the initial conditions. And then how does that really contribute to the radial position of the chromosome? I mean, I think that's an interesting question. We haven't really explored it yet.
Thanks.
Thank you for the nice talk. I was wondering also for the coalescence of the nucleoli, have you played with the physical properties of the chromatin around it, right? If you were to change its rigidity, persistence, length, and so on, your entropy barrier might actually change and you might vary the number of the nucleoli that you end up with.
You mean if we vary
your chromatin, yes.
the chromatin? Well, right. I mean, that's kind of what we did with the tumor, right? So in the tumor's case, the only thing we changed here is the interaction among the chromosomes, which are now parameterized with the tumor high-C data, right? So in this model, the interaction was parameterized from the normal high-C data. Right. So, but everything about the protein concentration, protein interaction, protein interaction, they were all kept the same.
I see. Yeah, I was wondering that with different parameters for your chromatin interactions in the top example, that you might actually shift, right, your number of droplets basically more to the right, for example, that you might get arrested several droplets, basically preventing them from fusing across the nucleus. Is that possible?
Sorry, could you refer that?
So if imagine in your system, so you start with 10 nucleolar organizing regions, right? And you start to generate those droplets. So as they are coming towards each other, so depending on the elasticity or persistence length of my chromatin fiber that I have, right? So the viscoelasticity of the network in between will be different. So basically, if it's more, let's just say, fluid-like, it will go easier together. If it will be stiffer, they might get like arrested separately. So I was wondering if you played with that.
Well, I think that what you said is exactly why we're ending up with a small droplet in the tumor, because they do have weaker interactions, so the network can actually relax sort of much faster, which sort of then allow. They would actually sort of there's more, I would say, entropic penalty for them to really get together, because now the network really prefer a more relaxed state, so that if you try to sort of imagine if you try to sort of constrain a more loose network, right? That would incur more sort of entropic penalty.
Yeah.
That's what we are saying.
And
but I guess I think what you're saying it's.
That you could have a state also with like a larger number of droplets, right, in the upper case. Because think if one would introduce, for example, heterochromatin now, and you would have regions.
Sorry, that's actually not our nucleoli. Those are the centromere clusters.
Oh, okay.
Yeah.
We should be speaking about the other slide where you had that. Yeah. But similar. So, okay, another question that I was wondering about, you mentioned when there was a nice student question about the droplets, why the small ones disappear. So you don't have a condition in your model that actually the NOR, so the rDNA, when the droplets fuse, actually has to come with it, right? Because in vivo, they do bring their rDNA loop with them. So if you have the larger nucleoli, they might have 2-3 loops you can see visible in them that originate from the initial nucleoli.
Well,
or is it like
we have a specific interaction between those protein and the NADS regions. So I mean, because if you look at the contact between chromatin and the nucleoli, it's not just those those specific transcription regions. There are actually there are a lot of more contact seem to be there. So we basically identify those regions from ChIP-seq data and just sort of say those all can interact with the nucleoli.
I see. All right, thank you.
Any last burning questions? All right. Oh, wait, we do have one.
Yeah, just to follow up to the previous question. So in terms of the origin of this barrier for two things coalescing, so I guess if you, so is it like more of an entropic effect or enthalpic? So what I understand is from your model, you do have like interactions between the polymer as well. So if you kind of, if you have two beads coming together, that means that it's stretching out the polymers behind it. So which means that the polymers have lesser configurations to sample, and that's more of an entropic effect. Whereas if these two nuclei are coming together, it means that it's also probably be breaking, sticky bonds between these two regions. So that's more of an enthalpic effect. So have you looked at both? And like, could you comment on which one maybe dominates? Or is it possible even to do that?
Yeah, no, I mean, that's a great point. Well, okay, so the way we know why it's entropy is because we can directly compute the entropy in our model, because that's why, we're working with sort of a computer simulation. We can calculate anything we like. And here, this is basically the entropy. You can see that the negative entropy actually increases, meaning the entropy is actually decreasing as we try to force the particle to be close to each other. So that's how we know it's actually mostly driven by entropy. And we also sort of did test that, if we sort of start to just remove the polymer connection, which shouldn't perturb the interaction, we also sort of don't really see this barrier. Everything is going to be down here and everything. Yeah.
Wonderful. Let's thank our speaker one last time.
Thank you.